Now, the regulation of pancreatic secretions, we're going to have to look at this over the several phases during the digestive process, that being the cephalic phase, the gastric phase, and the intestinal phase. The secretions and the regulation of secretions in the pancreas occur in a certain order. So I think the best way to do this is I will draw a very large acinar cell. And recall that the pancreas is a gland, and we are going to be talking about the exocrine pancreas. The exocrine pancreas is structured very much like the salivary gland, where we have an acinus and we have ducts that lead the secretions from the acinus down through the ducts as they collect and eventually empty through the sphincter of Oddi into the duodenum. We'll talk about the endocrine pancreas a little bit later when we get into glucose management and insulin secretion. But for now, let's just focus on exocrine pancreas. And the first phase that we're going to look at is what's called the cephalic phase. And the cephalic phase is, again, the preparatory phase. Recall the cephalic phase in the salivary gland was just beginning to stimulate the secretions from the salivary gland at the sight or smell of food. In the stomach, this also happens to start beginning the process of acid secretion through gastrin stimulation, and that's just to prepare the stomach for the anticipation of receiving food. Well, the cephalic phase also has some effect on the exocrine pancreas, and this is going to be through acetylcholine, again through the vagus nerve, and the acetylcholine binding to its receptor is going to, of course, increase the cytosolic concentration of intracellular calcium, and this is going to allow the fusion of vesicles with the plasma membrane. Now, what are in these vesicles? Well, these vesicles have the enzymes that are required for digestion. So they will include proteases, which will, of course, digest proteins. They will include lipases, which will digest lipids, fats. They will have glycolytic enzymes for carbohydrates, such as amylase. And finally, they will have nucleases for DNA and RNA digestion. So this makes sense. With stimulation of the vagus nerve in the cephalic phase, we get acetylcholine binding, triggering the cytosolic increase in calcium, which induces the fusion of vesicles to get the digestive enzymes into the lumen of the acinus and then transported to the duct into the duodenum. Again, in preparation for the anticipation of food showing up. So we have the enzymes already there. This is also important for the activation of proteases, because there is a specific activation sequence that is required for protease activation. And we'll see this when we talk about digestion, the actual enzyme digestion process. The other neuropeptide that does a similar function is GRP, which is gastrin releasing peptide. I didn't mention this when I talked about the release of gastrin. The control of gastrin in the cephalic phase is primarily done by the vagus nerve and acetylcholine activation. But GRP, gastrin-releasing peptide, is another hormone that also has an effect on releasing gastrin from D cells. In this case here, it's also part of the cephalic phase and does the same thing as just a little bit of fusion of these vesicles. Now, we're not talking about a full-on release of vesicles here because the cephalic phase, again, is the preparatory phase. The next step gets into the intestinal phase. This is where the real action occurs, is because when we get to the intestinal phase, we now have sufficient information that is going to cause the release of another hormone and cause a much greater release in the enzymes. The main hormone we're talking here is CCK for cholecystokinin. Cholecystokinin is a small hormone effector that binds to its specific receptor, and it has the similar properties of increasing the calcium concentration. Now this is going to cause a very rapid signal, fusion of lots of vesicles, and secretion of a lot of digestive enzymes because we're in the intestinal phase. And this is the real digestive phase. So we need lots of enzymes out there so we get a bigger signal. Now notice that all of these signals are all feeding into the similar pathway of increasing calcium. So we have the potential here of that same phenomenon that we saw with the secretion of the activity of gastrin and histamine, a potentiation effect, and that's what gives the amplified effect. Now what triggers CCK? We'll look at that in the next video. I'd like to touch on one more hormone that occurs also in the intestinal phase, and this is the hormone secretin. Secretin binds to its specific receptor, and secretin functions by increasing the cytosolic concentration of cyclic AMP as its second messenger rather than calcium. And the function of this is to increase the secretion of large amounts of bicarb into the lumen of the acinus and down the duct. Again, this occurs in the intestinal phase because the signal secretin lots of bicarb ion, and what is this going to do? This is going to neutralize stomach acid. The other thing it's going to do is increase the amount of water flow from the pancreas, and that will wash the pancreatic enzymes from the pancreatic ducts and into the duodenum. Now in the last video I talked about the secretion of water by the small intestine, and that was vasoactive intestinal peptide. And vasoactive intestinal peptide has the same effect as secretin, but it's not as pronounced or as strong a response as secretin. So these are the three phases. We have the cephalic phase, where we have a little bit of signal to start preparing the way by putting small amounts of digestive enzymes out. Then the next phase, the intestinal phase, is when we want the full push and full secretion of the pancreas, the exocrine pancreas. And so there we're going to have CCK is going to dramatically increase the effect of the second messenger calcium to increase dramatically the fusion of vesicles, releasing enzymes. And we also simultaneously want secretin and VIP activity in order to secrete large volumes of fluid that contain high concentrations of bicarb to neutralize the stomach. Now remember, all of this is coming out of the sphincter of Oddi into the duodenum, and in the duodenum is the exit port of the stomach. So as the acid is coming in from the stomach into the duodenum, the neutralizing bicarb solution from the exocrine pancreas is also mixing in here to dilute out that acid and to neutralize it. We want to take the pH back to between 7 and 8 because, again, the digestive enzymes work at their optimal pH of approximately between 7 and 8.